Okay, great. I'm sharing my screen right now. If there's a problem, please let me know because I can't see the interconnected forum right now. So presumably everybody can see it. I'm talking to you from Penn State University, right in the middle of Pennsylvania, at the tip of the Appalachian Mountains. And the building that I'm sitting in, where my office and lab are, is shown behind the screen here. It looks like a spaceship ready to take off, and we like that because we like to think of discovery as inspirational here. I'm going to talk to you today about the microbes' master plan to control sperm and shape generations. And I want to sort of set the context that when we think about microbiomes, a lot of microbiome scientists go to two anatomical systems first. They go to the guts of animals, or they go to the roots or leaves of plants. And yet, I would like to maybe influence your thinking today that, in fact, the reproductive microbiome has often more profound influences on host biology and evolution than those two that we commonly think about first. And to put the reproductive microbiome front and center as we think about the microbiome science's impact, especially in host-associated context. And so this is a story that begins 100 years ago, exactly 100 years ago. So it's just great to be able to talk to you all today with this history. The bacteria Wolbachia, the focus of today, was studied and discovered in 1924 in a paper by a graduate student and a PI at Harvard, Dr. Wolbach, hence the name Wolbachia. Now, their paper here has a sort of a prophecy stated inside of it, because what they were doing is they were ripping open the abdomens of a few insect species, including mosquitoes, looking into the ovaries, and then finding rod-shaped bacteria inside the ovaries. And to them, this was astonishing to think that there could be bacteria inside the ovaries of an animal. And they said this type of organism is one or more or less distributed throughout the whole group of arthropods. Now, that is a big claim. But in fact, because they had looked at a few insect species, they thought we could probably extend this discovery. And they were exactly right. About half of the world's arthropod species today, based on PCR screens, are now known to carry Wolbachia bacteria in the reproductive tissues, the testes and ovaries. Now, if you extrapolate 50% infection frequency or occurrence of the symbiont to the fact that arthropods are 85% of all animal species, and you do some number shifting, you end up with millions and millions of arthropods or animals that harbor the bacteria symbiont Wolbachia. This makes it the most successful bacterial symbiont in the animal world today. Now, how did it do that? That's going to become part of the topic today, and how can we apply it by extension? So we work on a number of different systems. You'll hear a lot about flies and Drosophila today, but this is my first love as a graduate student, Nisonia parasitoid wasps. They're about the size of a fly, and you're going to watch some mating behavior between a male and female here. If you were to take this wasp and cut open its abdomen and look inside the ovaries like Hertig and Wolbach did, you would find these Wolbachia bacteria. They're not too interesting to look at. They're small, micron size, semi-rod shaped. Sometimes they have multiple eukaryotic membranes around them to cloak themselves inside the cytoplasm of the animal cells. Now, if we stain them for fluorescence with fluorescent stain and look for Wolbachia, you can see right in the testes there are these red Wolbachia chock full inside the testes where sperm are made. And then in the ovaries, if we stain them with orange, you can see them right there in propagating as the eggs, the oocytes develop. And ultimately, these bacteria are maternally inherited. So the bacteria population from the mother's ovaries is what gets transmitted to the next generation. The father's bacteria does not get transmitted, but it is so important to the process of the spread of the symbiont that we'll talk about in a second. So in that spirit, with the fact that there is maternal transmission, if you were to look at this wasp embryo and stain in green for Wolbachia and in blue for the wasp DNA as it mitotically divides in the embryo, you can see these bacteria are well underway in replication, localizing to what we call the posterior end of the embryo. And this end of the embryo is cunningly where the reproductive tissues develop. So the testes and ovaries develop from this portion of the embryo. These bacteria, before there's a reproductive system, nowhere to be in the reproductive system by the time that begins developing these tissues in the larvae and pupae. Okay, let's go deeper. So if you were to keep pushing down into the symbiosis under electron microscopy, Michelle, many years ago, snapped some photos that to this day still are astonishing to us because she saw this bacteriophage portion of the Wolbachia cells. In fact, this is a bacteriophage portion busting out of a Wolbachia cell. You can imagine its membrane is here, and then the membrane is broken here where the phages are moving out, going to find a new Wolbachia cell inside this symbiosis. This is a healthy Wolbachia cell, so very granular in structure, a couple of membranes around it, an outer and inner membrane as we'd expect. But then right above it with those black arrows is a Wolbachia cell that's dying. And it's dying with a degraded DNA patch and membranes collapsing inward from the outer membrane and the inner membrane towards the inside of the cytoplasm. And the phage particles are making their way out. And even we can see these phage particles in the cytoplasm and tissue matrix of the reproductive tissues. This is actually in the testes of the wasps. And we see these phage particle packets now migrating around the tissue matrix, probably to find a new Wolbachia cell. So it's a tripartite symbiosis. And this becomes central to a lot of the biology of why Wolbachia is successful. Now, a lot of folks have come to learn about Wolbachia in the last decade because of its applications. There are many mosquito releases ongoing across the world, one of which is the World Mosquito Program. And about 2 decades ago, they took the Aedes aegypti mosquito vector of Zika and dengue virus, and they put in the Drosophila melanogaster Wolbachia by injection into these mosquitoes that don't have Wolbachia. And they were able to support the discoveries in flies that mosquitoes as well are resistant to RNA viruses that are pathogenic when they have Wolbachia present. And what that means is these mosquitoes now with the symbiont are immunized, in which the Zika and dengue virus don't replicate. Now, this is kind of magic in a microbe and a mosquito, because if you release this combination now into the wild, so these are shown in green here against the gray mosquitoes that don't have Wolbachia, and as they spread through the population through an adaptation that we'll talk about in a second, ultimately you can turn a gray mosquito population that transmits disease, into a green one that does not transmit Zika or dengue virus. This is happening across the world with wonderful success for human health. There are a number of towns and cities across the world in which we've seen declines in local dengue virus incidents when these Wolbachia carrying mosquitoes are released by the World Mosquito Program over periods of months of releases. Ultimately, the goal, of course, is to get to 100% reductions in dengue viruses. But these numbers are astonishingly impactful for transforming human health through Wolbachia symbiosis in insects. So how does this all work? How does Wolbachia sustain itself in a population? How is it spread to 50% of all the world's arthropod species? And one of the adaptations that has enabled Wolbachia to do these wonderful successes is a phenomena called cytoplasmic incompatibility. Now that's a mouthful. I'm going to walk you through it in this kind of Punnett square type format, where if you take a male with Wolbachia who's colored in, let's call that an infected male with Wolbachia, and he mates with an uninfected female, the embryos will die during early development, and you get few to no offspring in what we call the CI, or the cytoplasmic incompatibility cross. However, if those same males are crossed to an infected female with Wolbachia, offspring are produced at normal rates. And all of the offspring will have Wolbachia because the mothers, remember, maternally transmit the bacteria to the next generation. Even when the males are uninfected and they mate with an infected female, the offspring are infected. And so what this means is that the bacteria occur in twice as many offspring as do the uninfected cytoplasms without the bacteria. And that gives Wolbachia this extraordinary fitness advantage to spread itself through populations, to take over the arthropod world, and also to influence the success of these vector control applications, because the bacteria with this RNA blocking ability essentially spreads itself in the population. Now that CI death in the embryo happens because of a mitotic chromosomal failure in the embryos. So what you're looking at here is a father's and mother's chromosomes, just as they're about to undergo mitosis. when they meet in the fertilized embryo. But in a CI cross, you see something different. In the middle here is the paternal genome being shredded to pieces to create a dead aneuploid egg. And in some CI embryos, they can actually make it past this error, but several mitotic divisions haven't happened. And you can see these blank spaces where DNA should be, but doesn't occur. This embryo is under what we call mitotic arrest and won't develop any further after an hour and a half of development. These embryos will die as well. So the father then with Wolbachia is delivering an embryonic catastrophe for chromatin and mitosis that remained enigmatic both from a genetic and a mechanistic perspective until recently. So our question for a decade or more has been, what are the genes in the Wolbachia genome that weaponize this great pandemic of Wolbachia that occurs so widespread, and that also is directly leveraged for vector control, but without any genetic or functional basis known until some of this work happened. So it's been a century of work in the making, and I'm going to tell you the story of how we got there for the rest of the talk. So the first thing we did is Jason and Sarah in the lab did a multi-omic analysis from genomics of strains that cause CI and don't cause CI, and proteomics and transcriptomics. And to boil this down in a very short nugget, what they found are that 2 genes only strictly associated with CI, and we call those genes CI factors A and B. Now, for those of you that do genomic or multi-omic analyses, normally you get hundreds of candidate genes. So when we got 2 candidate genes, we thought we were either dead wrong because nobody gets 2 genes and they happen to be the right genes, or perhaps we were on the right track. Let me tell you a little bit more about that. So these 2 genes are called CypA and CypB, and they're localized in the prophage, so the inserted phage called phage Wolbachia. And these genes live next to each other in what's called the eukaryotic association module, which is a massive portion of the phage genome that presumably interacts with eukaryotic processes. And that in and of itself is an interesting story to think further about. The rest of the genome makes a typical phage: base plate, head, replication, tail, and so forth. Now, these 2 genes have a phylogenetic mirror. So they're divided into multiple types, and those types are concordantly diverging with each other, as if the CypA and CypB genes might be working together. Now, again, to make a long story short, since we're going to get into some of the mechanistic work today, we had a team of graduate students and undergraduates, as well as senior staff in the lab, take the cytoplasmic incompatibility phenomena and ultimately show that CypA and CypB can transgenically in flies replicate the entire process of cytoplasmic incompatibility. So CypA and CypB in males, when expressed and crossed to an uninfected female, causes CI. When CypA and CypB are expressed in the males, and then you cross them to a CypA expressing female, a fly transgenically expressing these proteins, well, then you get normal offspring, and those offspring are rescued. We did all of this without Wolbachia present. So we've synthetically engineered the symbiosis to its genetic fundamentals without the bacteria even being present. I'll show you the cross that really brought all that together. So what you're looking at here is the axis of embryonic viability, or hatch rates. And so a low hatch rate means CI, 20 to 30%, and a high hatch rate generally means compatible or rescue. So you can see in these wild type Wolbachia and transgenic fathers that they cause low hatch rates. That's the CI cross. And then this is the bread and butter. So this is when we cross those males to a CypA female, and then we get rescue. So what is a worldwide symbiosis? Because of cytoplasmic incompatibility, only has 2 genes necessary to make it all work. Now, under the hood there are domains in these proteins that became of interest to us. And we wanted to understand sort of what is the essential parts of the proteins that make the CI system work. And so Dylan, a graduate student, and Mahip, an undergrad, went to work on mutating sites across the proteins. And so when you see these labels, we're switching an amino acid to an alanine to mutate them, and then we're going to test the function. You can see that there's a number of color-coded domains that we'll talk a little bit more about in a second. To sort of condense this down, what we find is that there's a division of labor for CypA. The front half of the gene is involved in rescue and is necessary for rescue. The latter half of the gene is not. For CI, the front half, and actually the front 2/3 of the gene, are required to express CI. The latter tip of the gene or protein is not necessary for CI. Now, this is beautiful because it tells us that CIF-A has genetic capacities on the CI and the rescue side that ultimately determine that dual role it plays in the biology. For CIF-B, it only occurs, it only causes CI on the father's side. So everywhere we mutated it, it was found to be an essential region for CI, and that makes good sense in light of what we know as well. Okay, so once we found these genes and kind of understood their protein essentiality, we then wanted to ask questions about what's the mechanistic basis, the cellular basis of the CI rescue system. And you're looking at a beautiful testes staining of testes DNA and RNA here that'll become central to the story. And so let's get into the mechanism now. So this requires that we think about reproductive biology of the animal system. And in Drosophila, the testes kind of have this pinwheel structure with different stages of development. And ultimately, the mature sperm are at the end of this pinwheel on the inside here. Now, if you rolled out the pinwheel into a straight line, you'd see these stages of development from the germline stem cells all the way to the mature sperm. Our questions were, where and when do CIF-A and CIF-B occur? So what part of the testes are they in? And when do they show up? And then what, if any, are the paternal genome modifications as the sperm are developing in order to generate the CI system? And we call this generally the host modification model of CI because these proteins are wielding their action during the development of the sperm. Now, remind you that in a fly, which has Wolbachia, that Wolbachia has prophage, and that prophage has the eukaryotic association module, which in turn has these CIF-A and CIF-B genes. Now, here are the domain structures that we know of thus far. There is a nuclear localization signal in CIF-A. Now, this is kind of astonishing because remember, this is a phage gene, a phage-associated gene, now with a eukaryotic nuclear targeting sequence right in the middle of the protein. And this raises the question, does CIF-A go to the nucleus and maybe bring CIF-B with it? This is where Rim, or Rupinder, took over. She's an assistant research professor in the lab, and she developed and polished some antibodies for CIF-A and CIF-B that allowed us to see and test this hypothesis about nuclear localization of phage proteins in reproductive tissues. So again, your testes development is up here, and those names above correspond to the names here on where we're seeing CIF-A. So CIF-A occurs right from the beginning in the apical germline stem cell niche, all the way through to the development of these spermatid-looking or sperm-looking stages called spermatids. And CIF-B pretty much has the same nuclear localization, a little harder to see in the apical tip, but right after in the spermatogonia, we can see CIF-B associated with the nuclear DNA. And even by the time of the canoe spermatids, you can see that CIF-A and CIF-B are at the tip of the sperm heads, probably wielding some kind of action on those sperm. But it also doesn't preclude that their action could be occurring on the DNA and RNA of the developing sperm, which clearly have this nuclear affiliation. Now, this was done in flies by Rim, and then later she also did this with mosquitoes' sperm. And we can see in either Aedes aegypti or Drosophila melanogaster, the sperm have CIF-A and CIF-B as nuclear affiliated. Now, this is great because it confirms that this nuclear localization signal is likely taking its action to get these phage proteins there. In that spirit, we worked on mutating the nuclear localization signal to see if that disrupts the nuclear localization. So we have a wild type nuclear localization sequence for CIF-A that's transgenically expressed in flies, and then we just deleted it out of the sequence. And as you can see, the CIF-A in green no longer associates with these very young spermatids. That's because we disrupted that nuclear localization sequence. There's a phenotypic consequence, of course. So if it's not nuclear associated, then we expect the system to not cause CI. And indeed, that's the case. We get these very high embryonic hatch rates with the mutated nuclear localization sequence, but not with the wild type. So this really rounds out that CIF-A essentiality for nuclear localization and CI. Now, there's another aspect to the story about the development of sperm that we started to form hypotheses around. And there's a fundamental process in animal spermatogenesis from insects to humans called the histone to protamine transition. Now, this occurs in the latter half of spermatogenesis where you get these needle-shaped sperm formations. Before you get to that point, you'll notice the sperm cells are round, and they are round because the DNA of the sperm is wrapped around histones. And those histones are large proteins, and so you get these kind of circular shaped nuclear cells. On the other hand, when the histones come off at this stage and these smaller protamines come on, you'll notice the sperm take this needle head-shaped formation. And that's because the DNA wraps tighter around these smaller protamines than they do around the bigger histones. In fact, this green wheel here to reflect the protamines are multiple protamines. So not just one, but multiple small ones. And the DNA really loves to wrap tight around that, squeezes the head from a round to a needle shaped, and then we get our normal typical sperm. Now, in humans, infertility is often associated with a disruption, guess what, in the histone to protamine transition. So this seems like a likely course for similar actions of something that causes a sperm-egg incompatibility. This work has been done by RIM in both flies and in mosquitoes once again. And here are the spermatid stages with an antibody staining for histones. Now, when there's a no-CI condition, during this transition stage, the histones come off and we expect to see no histones. That's what we get. But in the wild type Wolbachia CI and in the transgenic CIF-A, CIF-B CI, we see a prolonged hanging on of these histones during that histone to protamine transition. This is the first indication that something in sperm development has gone bad because of the CI system. And on the flip side of that, the protamines have to be deficient now because the histones didn't come off, so the protamines never get to come on. Now, this is a stain called CMA3. It's a DNA stain that stains where the protamines bind. So if it's present, if it's a high intensity stain, that means the protamines aren't there and the stain is binding, as you can see in the wild type and transgenic CI. However, if the protamines are there, the stain can't bind, and that you get that light staining in the no CI control. So it's this type of data that allowed us to see that the histone to protamine transition has gone awry under the CI system. And moreover, if we use Drosophila mutants to knock out protamines and therefore create more of a deficiency in the system, we lower the amount of protamines in the system. What we found is that the CI strength gets stronger, meaning less embryos hatch on average than the wild type CI. So this connected a causation between the histone and protamine problems and now the ability of CI to take its full effect. Okay, so there we are. We've got this sort of late stage sperm development histone to protamine problem. We know that probably this is being set up by nuclear localization phenomena, but we don't know what happens early in spermatogenesis. And I want to talk to you about the rest of these domains because there are two domains in CIF-A that are RNA acting, putatively RNA acting domains. One's a transcription factor, one's an RNA binding domain. And then on CIF-B, there are two DNA aces that are putatively interacting with DNA. And this creates hypotheses that perhaps even before we get to the histone to protamine transition, the CIFs are modifying the central dogma biology of sperm development, RNA and DNA. Okay, so we undertook two approaches to look at this, both in vitro and in situ, right in the testes. First, in vitro. If CIF-A and CIF-B are in vitro enzymes that modify DNA RNA, we can test that by purifying these enzymes, giving them a DNA substrate first. So this was single-stranded DNA and double-stranded DNA, and then asking, do the enzymes purified out in a tube cleave DNA? And what you see here is that CIF-A and CIF-B cleave DNA relative to a no enzyme control and relative to an EDTA plus control, which means that EDTA stops the enzymes from acting. So whether you give it single-stranded or double-stranded DNA, CIF-A is acting as, and CIF-B are acting as DNA aces. And if we go now into the testes and look for DNA damage with what is called a tunnel stain for DNA nicking during the canoe stage of the spermatids, we can see in green that there's an elevated amount of DNA nicking and damage in the wild type and transgenic CI, more so than in the no CI control. So we have now a full spectrum of confirmation that these two enzymes are wielding activity against the DNA integrity of these developing sperm. Now, in terms of the RNA side, we did something similar with CIF-A, in which we were able to give it RNA substrate and show that CIF-A is cleaving RNA, CIF-B is not, and this is the RNA-only control, and the EDTA plus controls. If we go then into the tissues and look for what types of RNAs might be modified, RIM had this fantastic hypothesis of looking at a specific long non-coding RNA in the spermatocytes that was previously known to be involved in the histone to protamine transition of Drosophila melanogaster. And there was already a fish marker developed for it. So she went ahead, put it all together, and stained for whether this particular long non-coding RNA, important to the histone to protamine transition, is influenced by CIF-A. And what you see in the wild type CI and the transgenic CI is very little of this long non-coding RNA is produced early in the development of these spermatocytes. So this is before the histone to protamine transition, this kind of a regulatory long non-coding RNA. But in the no CI control, there's a good amount of this long non-coding RNA to do a proper histone to protamine transition. So now we have CIF-A as an RNase modifying very early a regulatory long non-coding RNA. If you then take mutations and ask what's happening to the long non-coding RNA, in the end of the CIF-A gene, you'll recall there was no effect of this particular set of mutations on CI. And as expected, there's very little of this particular long non-coding RNA. So this is a mutant that doesn't really cause an effect, so it has a CI capacity. However, in the RNA binding domain, in which we mutants ablate the CI function, so this would look like a normal compatible control, the long non-coding RNA is present there again. And moreover, if we then march into spermatogenesis, we can show that these two mutants strongly correlate with the expectation of histone retention when there's CI and protamine deficiency when there's CI, an elevated protamine deficiency and an elevated histone retention. So the long non-coding RNA is setting up this histone to protamine defect that we see in CI. Okay, the final step is RIM is developing a kind of window into the key master switch of CI. And so looking at this process of spermatogenesis, we thought that if histone removal is essential to the process, then if we just stop histone removal with a chemical inhibitor, we might be able to recapitulate CI. So this is the control crosses. What we're looking at are flies with normal Wolbachia from melanogaster that cause CI and flies that have rescue. This is the next two crosses in which the flies don't have Wolbachia, but they were treated with a chemical inhibitor. So it lacks histone retention, presumably, it has histone retention, presumably because of the inhibitor, and we recapitulate that CI phenotype, which importantly is rescued by the females that have Wolbachia. That's an essential part of the process, telling us this is bona fide CI and rescue. And if you look at the histone retention, as expected, when you put in the inhibitor, the histones are retained, and when you don't have the inhibitor, the histones come off. So this completes then the pathway from genes to proteins to mechanism to now small molecule disruption of the pathway that allows us to recapitulate cytoplasmic incompatibility to degrees that are somewhat like the system seems simple, even though it's a very complex phenotype. So here's a summary. Flies, half of the world's arthropods have Wolbachia. Their genomes are tiny because they're endosymbiotic. Inside those genomes are prophages. Inside those prophages are eukaryotic interacting genes, including CIF-A and CIF-B. And those genes have domains very important to the process of how these phage genes manipulate eukaryotic nuclear biology in the reproductive tissues. So ultimately, we can boil this down to a cascade of catastrophes during testes sperm development. The first hit in the cascade is the long non-coding RNA depletion in spermatocytes. The second hit is histone retention. The third hit is DNA damage. The fourth hit is protamine deficiency. And so all of this is now leading to this cascade of host modifications that cause the cytoplasmic incompatibility phenomena. What this all comes down to is that we have remarkably prophage-encoded enzymes invading developing animal sperm nuclei. That this leads to an altered regulation of some of the dark matter of the genome, the long non-coding RNAs, that causes in turn chromatin integrity problems. And this is the first example of a symbiosis in which nuclear targeting phage proteins impact eukaryotic gametogenesis, sperm development in this case. If we zoom out to the big picture, we understand that there is a global symbiont of gigantic proportions in the animal world. A lot of that boils down to a very simple genetic toolkit with CypA and CypB from the prophage region. This is now being used as a technology to control mosquito-borne diseases by many companies across the world, including a spin-off company from Google called Verily, a Kentucky company called Mosquito Mate, and the World Mosquito Program. A lot of this is done with robotics. This is actually a van, an autonomous van that drives around with a roll-down window and robotically releases mosquitoes to control local cases of Zika and dengue. And from a fundamental textbook perspective, and I didn't get a chance to talk about this and would be happy to, CI is a factor that influences the evolution of their hosts in major ways, including sometimes the evolution of reproductive isolation and speciation. So with that, I will stop the talk. I'll take my slides down and be happy to entertain any thoughts you might have about this system and how it's being applied across the world. Thank you for listening.